evening, everyone. So I welcome you all to the 25th lecture of Archaeological Science Center webinar series. I'm Unka Padi, a PhD scholar here in IIT Gandhinagar. So this is the third year of our webinar series, and for the 25th lecture, we have Professor Sharada with us, who will be delivering her lecture on human-animal relations across time and space, case studies of recent approaches, and novel perspective. So I'd like to begin by introducing Professor Sharada Channarayapatna, who is an assistant professor in archaeology at the Archaeological Science Center in IIT Gandhinagar. She has 2 masters and 2 doctorates awarded by Deccan College, Pune, the University of Ferrara, Italy, and National Museum of Natural History, Paris, France, respectively. Specializing in ancient Indian history, culture and archaeology, and European quaternary and prehistory. Prior to this, she completed her bachelor's in history, economics, and sociology from Mysore University, and she has been the recipient of the Erasmus Mundus Scholarship, the Nehru Trust Small Study Grants of Independent Projects, the Indian Council of Historical Researchers Fellowship, and more recently, the DST SERB's Power Grant. She has, in the last 15 years, analyzed faunal remains from more than a dozen sites in India and Europe to delineate the site's prehistoric and protohistoric animal-based subsistence strategies and the taphonomic trajectories. So I would like to request Professor Sharada to please deliver the lecture. Thank you.
Thank you, Unka, for this very kind introduction. I will now share my screen, and please let me know if it's all right. And is it okay?
Yes, ma'am.
All right. So I have titled the talk as Human-Animal Relations Across Space and Time, because today through this talk, I hope to make you time travel with me across 2 different continents, Europe and Asia, and 2 sites specifically. One, a very old 5,83,000-year-old site called Isernia, and another site from the Harappan civilization, 2 sites, in fact, Diravar and Farmana from the protohistoric time period. And these are 2 special case studies because I wish to concentrate on a few methodologies and the possible interpretations that can arise out of the employment of these methodologies, and what kind of research questions we will address in order to understand this relationship. So very first slide, I have put a collage of different aspects of this subdiscipline of archaeology called archaeozoology, which studies the human-animal relations. You can see a variety of pictographic depictions of how we show these relations, how we interpret them, in which ways we can depict them. So I'll now move to a very general introduction of what is this subdiscipline and what do we exactly do, what's the scope of our subject. So archaeozoology or zooarchaeology is the study of animals in archaeological contexts. The primary goal is to shed the light on the relationships between humans and animals from very different time periods, cultures, and regions, and the consequences that these interactions have for both humans as well as the environment. And we know that animals are very much a part of this present day as well as the past or paleo environment. And along with animals, plants, and the physical aspects of this environment, such as soil, climate, temperature, etc., we are basically trying to reconstruct the human and paleo environmental interactions, a part of which is that between humans and animals. So what we do is we analyze the hard parts of animals like bones, teeth, shells, exoskeletons, which are recovered from archaeological sites of these animals. And we don't always just study one group. We can look at mammals, birds, fishes, reptiles, amphibians, although there are some specialists who prefer to study just certain types of animals like fishes, for instance, or small mammals or invertebrates, and shells, for instance. So we not just look at the hard animal parts, sometimes we also have to corroborate this direct evidence with other sources of information, which can come to us from historical documents, ethnographic studies. There could be iconographic details, or even paleofecal or coprolites, what we call, such as owl pellets, which can also contain animal bones. And so we can look at their composition in terms of their chemical ingredients, the isotopic signatures, or the genetic makeup of both these human and non-human animal remains. And we ultimately try to reconstruct these human-animal relations. So we deal with two types of sources, direct and indirect. Direct is mainly the actual tangible materials, like, as I said, bones, shells, teeth, etc. They are the primary evidences for us. But we also have indirect sources. As I just mentioned, there could be visual evidence, there could be textual evidence or oral histories, and we use them for corroborative purposes. So our play field, our main area of work is in these human habitations or past human settlements. And we know that humans have interacted with animals for various purposes, a wide spectrum of roles and significance that they have held for us, either as food or for non-food usages. In case of food, we have wild and domestic animals. In case of non-food usages, we have used them as working animals, we have traded them. Sometimes we use them as raw materials, and we also keep animals with us as pets, and they become commensal beings. There is a symbiotic, mutualistic interdependency that has built over millennia and centuries. In this human habitation containing remains of these animals, there can also be other animals which we don't necessarily consume or use for these other purposes. There could be scavengers who enter the site, like hyenas or other rodents, for instance. They can be accidental inclusions, chance inclusions of animals that have died near the site and have got incorporated with the faunal assemblage. Ultimately, what goes into the archaeological context is the food refuse, as well as the animals which die and are thrown away into refuse pits, or there could be accidental inclusions, and they get buried over in the soil over a period of time. And this is where this picture gets complicated, because there are lots of different actions of cultural and natural agents which modify these bones. And finally, when we recover them from the archaeological sites through excavations, we are left with a very small number, probably just a sample of what originally would have formed or got deposited in the habitations or settlements. So, for example, here you have a big, beautiful, majestic looking bison, a live one. And at the end, what comes to us is just a bag of bones, dry, dead, and we have to try and bring life back into these bones and interpret them. So these are some possible roles and ways in which we interact with the animals in the past. I have tried to list out as many as I could think of, but there are a few that I've left as question marks, and I'm sure you will also have your own contributions to it. I may have forgotten some. So we have depicted animals through beautiful rock art in caves and rock shelters. We, of course, use them as a source of our dietary protein and nutrition in terms of food. We have used them as clothing, their skin, their hide, their body parts, the bones, antlers, etc., have been used as implements. We have fashioned objects out of them. We have used them for medicinal purposes, also as labor, either in the agricultural fields or for trade and transportation. There are ornaments made out of them, manure. We have incorporated them into gambling, sports, and warfare. We have developed a very beautiful relationship with them in terms of companionship and stress relief. There are lots of stories and beliefs that revolve around them in terms of folklore. And of course, we have deified them. We have raised them to this divine status at times. So almost every part and every aspect, both tangible and intangibly speaking, they are a part of our lives. And this relationship is a very beautiful, primeval, at times not so beautiful. It's a checkered relationship that we have. And if we go back to the Pleistocene times, it's more of a prey-predator kind of relationship. But once we enter the last few thousand years, let's say 10 to 12,000 years, when we ended up settling down and became agriculturists and gave up our hunting, gathering lifestyle, we became more like their protectors and not their predators. So the direct sources for us are mainly macro remains, like these bones, teeth, antler, shells. Otoliths are these small ear stones that we find in the fish's ears. So these are the otoliths, and shells, antlers. Here we have the exoskeletons like made of chitin. There are stomach stones, feathers, or impressions, like this Roman pottery here has the footprints of dog or a puppy on it. There are also a lot of objects made out of them. There can also be micro remains, which are not visible to the naked eye, but are equally important, like the biomolecules, DNA, amino acids or proteins, as well as lipids present in pottery and other sources, which can help us to delineate some of these relationships. So as I mentioned before, I'll be taking you across the globe from one side to another, from Europe to Asia. So on this chronocultural timeline, I have highlighted these sites here. The first is Isernia La Pineta. It's an Italian a Lower Paleolithic site, around 600,000 years old. And then we'll move across to the protohistoric period or the Harappan civilization in India. And we will look at these two sites, Girawar and Farmana, which is around 4,500 to 4,000 years old. And so I will first begin with the first case study, which is Isernia La Pineta, where I intend to show you what a European prehistory site looks like, what are the animals that are found, how do we think they were exploited or utilized, and what happens to their remains ultimately on the site. And here, the main aim was to understand the taphonomy behind these remains. That is, once we know that the humans have exploited them, so what happens once these remains get buried in the soil, and how does the depositional environment, the formation processes act on these remains, and how they can create biases in our inferences. So the site Isernia is in south central Italy over here, and it's the second oldest site in Italy after Nota Chirico. And it has, after 40 years of excavation, finally yielded one single human remain, which is the tooth of a young boy. And I'll show that to you. So it is found in these continental Pleistocene deposits, very complex geography, geomorphology in this area, comprising river, lake, runoff debris sediments. There are also tectonic movements and volcanic tephra that has got deposited over time. There's a volcano nearby called Rocca Monfina. And it is one of those sites which gives you non-Acheulean lithic finds. So there are no bifaces or hand axes, but there are other types of lithic tools. So this map here shows many different sites in very important sites in Southern Europe and Central Europe. And Isernia is over here. So it has a complex stratigraphy. There are 5 units which are called stratigraphic units, unconformity bounded, and they are, as you can see, they are made up of different sources. The oldest U5 is lacustrine lake sediments with gravel. Then comes travertine, which is the limestone, and it forms the bedrock. And then comes these paleo sediments, paleosols. There is sand, gravel, and this unit in particular, this unit 3, comprises all the archaeological evidences. And it is overlain by sands and gravels, and finally gravel, sands, and intercalated tuffs, the volcanic deposits. So this unit 3, I have zoomed in here because that's where the archaeosurfaces are located. They are 3C, 3A, 3 col, which is a short of 3 colluvium. And over them, there are several cuts and paleosols. And we are mainly interested in this 3 colluvium. That's what I'm going to discuss today. And there's lots of different dates available for these sites. And so there is biostratigraphy, the use of faunal remains to understand the age of these different layers. They have done paleomagnetic dating. There has been argon dating, potassium argon dating. So all these ESR of the tooth enamel, electron spin resonance, and certain dates have been sort of locked in for these important paleosurfaces. So the site, the area that we are interested in comes between somewhere between 600 to 583,000 years ago, 5 lakh or 6 lakh years ago. So this is the human tooth, which was discovered after 40 years of excavation. And it's the central lateral incisor of a young boy. And this is the archaeosurfaces. Very rich in archaeological finds. So I'll show you quickly how the site looks like. This is the main floor, or the living floor, or the excavation floor here. And there is a big canopy or a superstructure built over it to protect it from natural environments. And then there's a small passageway which leads to the museum. Here's another view of the same site. And this is inside the museum where most of these archaeosurfaces have been replicated and molds have been made of them and displayed, and several aspects like the stone tool making or the the animal butchery processes, they have been replicated in the museum. There are also these life-size animal replicas. For example, here we have the ancient elephant, the Paleoloxodon antiquus from the site, the straight-tusked elephant, which is usually found in the Pleistocene time period, early Middle Pleistocene in Europe. This is the excavation that's going on. This is how it looked like when I was attending the excavation. And you see people are lying down on either wooden planks or these sandbags and very carefully removing the finds. And the entire site is divided into two sectors, and it was actually discovered in 1978 when this highway was being built next to it between Naples and a town nearby. And the person who found them, Albert Solinas, he reported the finds to University of Ferrara, and the professors there immediately realized that it was a very interesting, very rich site, and they stopped they diverted the highway construction and they started the excavations. So the site is divided into two sectors. Sector 2, which lies north of the highway, is not very rich, but the southern half, Sector 1, is very rich in finds. And they are divided into 1 by 1 square meter trenches or working areas. And in between, you'll see these little squares that have been left behind and not excavated because they show us the stratigraphy, the vertical layers of the site. So here's another view of this. You can see a lot of bones and small pebbles and flint and tools. These are the skulls of bison. There are lots of teeth and tusks of the elephant and antlers of deer lying around. These are all very wild animals who are most probably, some of them are the ancestors of the present day animals like bison and horse and deer, etc. The finds, which are quite big, larger than 2 centimeters, they undergo a lot of restoration and conservation processes. They are put together, they're refitted, and adhesives are applied on them to keep them stable. And then they go for analysis. The smaller finds are retrieved through these buckets of soil, which are washed and then sieved, and then we sort them out. This is a typical methodology that is followed in almost all the excavation sites. But the interesting thing about this particular site is every single find is coordinated. It is 3D coordinated, and it is immediately exported into the GIS, and we have a visualization of where the finds are coming from as the excavation is happening. So everything is very well documented in this site. So the major finds include this limestone, and there are several flint and limestone tools, cores, debitage or waste products. There are also a lot of microvertebrate remains which tell us about the environment. So the site has a Middle Pleistocene stone tool assemblage with direct percussion bipolar techniques, and there are very small in size because they're mainly flake tools. And the complete chain operatoire, the whole process of creating these, manufacturing these tools is available. So we know that there was in situ knapping happening at the site. And several useware studies have shown that these tools were used for butchery activities. So the climate, as indicated by micromammals, shows that the site was slightly cooler than the present day. And so this is the 3 collovium layer, which I was interested in, and I studied the faunal remains from here. So these are the squares, 1 by 1 meter square area, and the number of remains from each square is indicated here. And so totally there are 15,000 and above finds, out of which 6,856 were bones, and the rest were tools or just blocks of limestone and other river pebbles. So, and this is what the surface of 3 collovium looks like. Very, very rich in finds. So once the preservation and conservation happens on the site, what I did was to immediately start analyzing them for what species they are, what part of the body of that species they belong to. And I entered the data in this MS axis. And I used some kind of codification so that if anyone else studies the site after me, they could follow the same codification. And it's very easy to enter data and quickly import it into GIS. So all these aspects of each find were recorded, and the context and the coordinates. And of course, each find was given a GIS code. Several macroscopic observations were made, such as I told, the species and the skeletal element. Then if they were fused, not fused, and different measurements were taken, the shape, the refitting, contour, the state of preservation, for instance, all these were recorded. And microscopically, I looked at the taphonomy, that is the bone modifications that were visible to me under the microscope, like trampling, rounding, marks of roots on them, rodent or carnivore marks on them, any indication of whether these bones had been impacted by the humans themselves. So we call it anthropic or anthropogenic modifications. And non-anthropically, whatever I could find, all of them were recorded. And then finally, a few statistical tools were utilized to find out the richness of each of these species in relation to the other species at the site, and what kind of animals, in this sense, what age groups these animals belong to, and whether the entire animal had been killed and brought to the site or only parts of it had been brought. So this is called differential transport of the kill. And then finally, all this information was fed into the GIS, and several queries were put, and several thematic maps were created based on different variables. And I tried to look at the concentration as well as dispersal scenarios that emerged, and what was this 3D or 2D map? What were these 3D or 2D maps telling me about the site formation processes, and how the faunal remains had got affected because of them? So these are some of the animals. I think many of us are not familiar with them. These are really old wild ancestral species, so belonging to the early and middle Pleistocene. So we had 4 orders, 7 families, 11 genera, and 10 species identified at the site through all the layers, not just 3 colluvial, even in other layers. So there's bison, and I've arranged them in this decreasing size order, not to show their actual sizes, but the relative abundance of these animals at the site. So bison are present in very large numbers, the way cattle, buffalo are present in large numbers in Harappan sites. Similarly, bison was their preferred hunt and preferred source of meat and other raw materials like bones or hide or skin. Then comes the rhino, the 2-horned rhino, and then the carnivore bear. And finally, we have the straight-tusked elephant. So as we move from bison to elephant, the number of remains of these animals are lesser and lesser in the faunal assemblage. And in case of bison, rhino, and bear, we have good representation of almost all body parts. But when we come to elephant, they are mainly the tusks or the head portion or the teeth of these animals that are present. So there seems to be some kind of thought that went into what kind of body parts to exploit or bring back to the site from the kill site, if the kill site was very far from the place where they had encamped. And then we have leopard, wild boar, hippo, and also giant deers. These are deers which have very large antlers, extremely large ones. And then we also have red deer, the tar, which is like a mountain goat and lives on higher altitudes. Then there's fallow deer, which have slightly smaller antlers, and then the roe deer. So as you can see, these are very mixed set of animals. They don't really come from the same kind of biome or ecological niche. They live, they survive, their habitats are quite varied. For instance, the elephant prefers slightly open or less forested areas, whereas the tar lives on very high mountains, like a steep area. The bison prefers open landscape near waters, so they move in large herds through the landscape. So obviously what it shows is that all these animals represent a very mosaic landscape. So this is an artist's impression of what Eternia could have looked like 5 lakh years ago. So you see a kind of a mosaic a mix of different steppy areas, flat areas. There are sloping areas, there are forests, these marshy, swampy areas where rhinos, hippos could have lived. and also large areas, open spaces, because most of these animals are megafauna. They are big sized, so they need open spaces to move through. And there are lots of birds and small mammals. And so they represent both temperate as well as a temperature, a kind of a climate that is slowly cooling down, and the temperatures are lowering over time. And another artist's impression of what the humans at the site could have been doing. So they are, as you can see, settled on these small flat spaces where they are butchering the animals and exploiting different parts of them, like meat, marrow, sometimes even the brain, because the skulls have been crushed. So how do I know that the humans were exploiting these animals? So we have to look for those signs, the cultural or the anthropic signs on the remains. So here I have shown two examples, that of bison and rhino, and we'll see through this skeletal part representation that most of the body parts are present. Now, that depends a lot on how these bones were processed and how sturdy they are in terms of their density or nutritional value or their size, and what has happened to them after they have got deposited. So relatively, teeth are very well preserved, for instance, or horns are well preserved. And the other bones, which are very rich in meat, they have not survived very well. These are some bones like the vertebra or the long bones, and they have a lot of cut marks on them, which were made by these small stone tools. Similarly, the rhino is also well represented by different parts of the body. And you'll see that these cut marks are very faint, and they have to be analyzed under the scanning electron microscope. And why we know these are cut marks made by stone tools is because they have this characteristic V shape in the section, and they have tapering ends. And inside these striations, we will see microstriations, smaller thin lines, and characteristically made by stone tools. So if the same mark were to be produced by a sand particle or a rock rubbing on the bone, they would leave slightly different marks. So here are some more pictures of those cut marks. This is a scanning electron microscope picture, which shows these deep incisions. Here are some more pictures. Sometimes we also have these scraping marks. So an animal, when it is butchered, it begins with skinning the animal, and then the muscles are removed, and there's a lot of scraping on the bones to remove the slimy part of it, which we call periosteum, a very thin layer of muscle or fat or tendon that attaches onto the bone. And then the ends of the bones are typically cut off to disarticulate large chunks of meat. And then finally, we have these thin cut marks to remove the periosteum or the leftover muscles. So all stages of butchery by one of our human ancestors is definitely present on most of these remains. And that's why it was quite evident that humans were definitely there on the site and exploiting these animals. But in many of the European sites, the first debate that you will hear about is a confusion as to, or a debate about if humans were active hunters, or were they confrontational scavengers, or were they passive scavengers. So it is said that in most cases, probably it was the predators, the large carnivores, that would have the primary access. They would kill these prey, and then they would consume most parts of it. And then the humans would arrive on the scene and consume the leftovers of the animals. And with the kind of tool technology they had, and the kind of landscapes they were living in, competing with these other carnivores as predators, it would not have been possible for them to hunt actively. And otherwise, they could have driven away the carnivores from the kill site, and they could have feasted on the remaining body parts. So there's this ongoing debate is there for almost all the European prehistoric sites. And in fact, for Ysenia, Jean-Philippe Prougal in 1990s had already said that probably these accumulations could have been produced by lions, and that humans had secondary access to these animals and their body and their meat and body parts. So I wanted to understand this better, and I found a methodology to employ here. It's called long bone fracture and fragmentation analysis, where I looked at the different bone fragments in terms of the kind of breakage they had undergone. So firstly, I described the different planes. So for instance, this is a long bone, and there are one, two, three, four planes to it, each of them broken in a different way. There's an oblique fracture, longitudinal, or a transverse fracture. And most of them have they have a certain angle, outline, and edge to them. Here are the descriptions for it. And the shaft or the main part of the bone can be recorded in terms of its circumference and length. Now, there are different possibilities which will arise, and there are numerical values given to them. So if a bone has angle of 0, outline of 0, and edge of 0, the total score of these three factors would be 0. If it had angle of 0, outline of 1, and edge of 1, the total score for this would be 2, 1 plus 1, 2. So if you look at all the permutations and combinations, there would be either 0, 1, 2, 3, 4, and maximum of 6. So maximum of 5. So, and also if you look at the edges, sometimes they're not very straight. They would have undergone different types of fractures. For example, there is a Y-shaped edge or a spiral edge or a perpendicular smooth edge like this. So for many different bones, I recorded these different criteria. And I wanted to understand what it would mean in terms of whether the bones had been modified by humans or carnivores or some other agents. So in layer 3, I analyzed 811 remains, and there were we saw that most of these are long bones, and there are many diaphyses. That is, these are the diaphyses, the central part of the bone. And in layer 3, again, we saw that most of the bones are very small. They're short. They have been fragmented. So their lengths are very short, and the large-sized elements are very few in number. So in terms of their fragmentation along the length and the circumference, most of them, again, were very fragmented bones. They had very short lengths and very short circumferences. Now, this is important because if the same assemblage were to be created by carnivores, the graphs would have been completely opposite. So these are bones that have been modified by carnivores. As you can see, the ends have been chewed off, but the circumferences are always more or less complete. The entire bone is, the circumference is preserved, and the lengths are much longer. But if the same bones were to be modified by humans, they would have these kind of fractures, a lot of different impact points and detachments like flakes, etc. And finally, what would emerge is that according to the angle, outline, and edge, the bars would have been higher, indicating human breakage. Then the bars would have been higher for oblique, spiral edges, smooth edges, for instance. And if the bones had been modified by carnivores, the bars would have been higher for variable angles or intermediate outlines or very jagged edges. So clearly, again, the assemblages were created mostly by humans because in very few cases we had the variabilities. There was a mix of characteristics, or they were completely not conforming to the criteria for anthropic or carnivore. They could have been post-depositional fractures. But definitely we had very few instances of carnivore damage, and those were mainly evident in the form of these different tooth marks or chewed and gnawed appearances of the edges. Most of the bones appeared in this fashion with lots of breakage and lots of detachments and cut marks as well on them. So finally, when I added up the scores for all these different planes of fracture, I saw that most of the values were between 0, 1, and 2, which indicates anthropic human activity on the bones, human intentional fractures and breakages. 3, a mid-range value, indicates a mix of both human as well as carnivore or post-depositional fractures and breakages. The rest of the 3, 4, 5, and 6 scores, they indicate more of post-depositional factors that would have broken the already bones that were already modified by humans in the past. So this kind of cleared my my confusion as to what was the main agent behind the accumulation of this faunal assemblage at Isenia, especially in 3 colubrium. So now I wanted to understand the distribution of these remains. So for this, we employed a GIS. I did not do the GIS part. I collaborated with someone who had developed the software and the programs for this purpose at Isenia. And so we created these 2D maps, which show that the faunal remains are quite, distributed over the site, but in certain parts, they're more densely concentrated, like the southern part here, or this is the southwestern part, but the central part is less, homogeneously distributed. And if we look at the cross section of the site, it seems as if the site is slightly dipping or sloping from the north to the south. So here are some more images of what the 3D cross section of the site looks like, of different layers, not just 3 columnium, of also 3A and 3C. And we see that there's a slight slope, and then there are areas where there are heavier concentrations and areas with less dense number of remains. So these 2 maps indicate the distribution of unidentified remains and the distribution of faunal remains of the major species like bison, rhinoceros, elephant, and bear. And again, we see similar patterns emerging where we have denser concentration here and lighter concentration towards the center and north. And so what does all of this mean? Similarly, even for anthropic fractures, we see more of more evidence towards the southwestern part and less towards the other areas. So these maps indicate that if at all this was in text site, then the humans were definitely carrying out most of their butchery activities towards the southern and southwestern part of the site. And the debitage or the waste was mainly being thrown in the other parts of the 3 columnium layer. And because of the dip or the slope, it seems as if over a period of time, had there been, say, surface runoff or erosion or debris flow or a colluvium sliding downwards, then the heavier elements seem to have settled down at the bottom part of the site, and the lighter elements, the smaller bones, they seem to have still, remained very randomly, haphazardly over the rest of the site. So the next question for me was to understand how badly were these remains compromised in terms of the post-depositional factors. So we know that humans have butchered them, discarded them. Now what happened to the remains? So clearly these 3 agents, weathering, erosion, and exfoliation, had impacted the bones the maximum among all the natural agents. So I decided to give them, decided to codify them again. So if these, the impact of these agents was present on the bones, it was numbered as 1, and if it was absent, it was 0. So, and I also looked at if they were, how badly they were affecting the bones, if they were acting individually or in combination with each other. So in 898 bones, they were totally absent, all the 3 factors. So a bone would have looked something like this here, which means it is very well preserved. But if all the 3 factors are there, which is 1, 1, 1, that combination, there are 623 bones, they're very badly preserved, extremely damaged bones, and this is how they would have looked like. So in between, we have only weathering or only erosion, only exfoliation, and the different numbers for them. So the next step was to visualize them on these 2D maps. So this first map, frequency map that was generated, shows the relationship between the faunal remains, which are altered by these agents in black color. So within each square, you'll see a pie chart, and you'll see the black and the light gray color. The black indicates bones which have undergone these damages by these 3 agents, and the unaltered remains are shown in light gray color. So we quickly, we can visualize that this part of the site was very badly damaged. The bones from here were compromised, and especially this square here. But these areas here, the south, the western part or the southern part or the southwestern part, are relatively less damaged. And another map shows the frequency of the faunal remains, which are altered by all the 3 taphonomic agents again. But within each square, you will see there are different colors. So we have indicated not just the number of remains that are coming from each square, but also a pie chart within that square, which shows how many of these remains are affected by exfoliation, weathering, or erosion. Again, it pointed us to the same scenario, that the central or the northern part are more compromised, and they are badly damaged. And there are also more number of remains coming from this area. And the southern part, southwestern or the western part are relatively better preserved. So combining all these factors together, what appeared to me was that because of the slope and the dip and the size of the elements and the different modifications they had undergone, it seemed as if the elements in the central part, they were very quickly, they were actually exposed on the surface for a much longer time, and they were not quickly buried as this layer was forming. But the remains from the other parts of the layer, they underwent a very quick burial. And that's why they were not exposed to these agents, and they did not undergo so much of modification. So I'll come to the second case study, which is from the site of Girawad and Farmana. And here again, I was looking at wild animals, looking at their spatial temporal distribution. But the only disadvantage that I had at these sites was that I did not have the 3D coordinates for each and every faunal remain. And that's why I could not use the GIS at the site, but I had some different types of analysis that could be employed here, and which could give me similar insights into a site of a much later period and much different culture. So moving to Asia now, South Asia in particular, in the northern part of India, in Haryana, I looked at the faunal remains from the 3 Harappan sites of Farmana, Girawad, and Mitatal. I will only be talking about Girawad and Mitatal. Girawad is early Harappan site, and Farmana is a mature Harappan site. And these were the primary sites for me. And I analyzed the faunal remains, and that's why they were, I could do different types of analysis, and I had lots of different variety or nature of data. But the other sites that I have marked on the map, these are the secondary sites for me. There are also Harappan sites like Bhagwanpura, Mirzapur, Burj, Pirana, Kunal, Masoodpur, Rakhigadi, etc., Alamgirpur. But these, I had the data for these sites from secondary sources such as publications, and of course, through personal communication with Professor Joglikar, who was helping me to carry out these analyses. So here again, in these sites, almost 5 lakh years later, we also see that humans, although by this time have domesticated several animals, they are inhabiting large settlements, they are cultivating crops, and their relationship with animals have changed quite drastically since we are now in the in the middle part of Holocene. But they also continue to interact with wild animals, but in a different way. But when we look at the Harappan sites and their faunal reports, we are usually, we encounter sentences saying that we are given a laundry list of wild animals, and we are told that they were, they played some kind of role in the lives of the ancient Harappans, but we don't know exactly what was this interaction. Were they being hunted? What happened after hunting? Was there meat distribution? Why do we have very few remains of some wild animals where other wild animals are better represented? And what kind of environment do they represent? So these are different questions that are very rarely answered in the context of certain Indian sites. So I wanted to explore these themes, but in a very different chronoculture and geographic space. So the first site is Girawad. I'll introduce the sites to you first. So Girawad is in Rohtak district, and it is a single culture site. Most probably, even if it had multicultural layers, the top part of the site has been destroyed completely because of cultivation. And it is 8 hectares in size. 41 trenches were taken across the excavation plan, and then 13 pit dwelling complexes were excavated here. You see them, each complex is denoted by this gray kind of, not really a circle, but gray areas. So each one is a pit dwelling complex, and 13 of them were excavated. And within each complex, there were many other smaller pits excavated, 78 in all. The ceramics were very similar to the Hakra ware. And the site has been based on relative chronology dated to 4th millennium BC, based on the ceramics. And we have a lot of different artifacts coming from this site. And so here is the excavated area, a general view of the site and the different pits. So what are these pits? Why have I given them different colors? Now, the excavator, after going through the contents of each of the pits, saw that there were different, probably the pits had different functionalities. So, for instance, some were garbage pits, some were dwelling areas, some indicated some kind of activity area, others were pottery kilns. For example, we have a picture of pottery kiln here. We have a garbage pit here, which is full of bones, and some were the round bases of pots, some were storage areas. So I have color coded the different activity functional areas across the site. And the next site is Farmana, which is again a multicultural site, but unfortunately it has also been damaged due to agricultural activities. The top part of the mound has been flattened off. And probably it is around 18 hectares in area, very large site in Rohtak district, again in Haryana, very close to Girawat. There is an associated cemetery, which is situated about a kilometer northwest to the habitation area. And if you look at the entire site, it's mostly the central part which has survived very well. And that's where most of the excavation happened, which has 2.5 to 3.5 meters deposit. There are 3 localities. The central part is locality 1, and north to that is locality 2, and further north, there is locality 3. So index trenchers were taken from all these different localities, 7 in all, and they revealed almost 12 to 13 layers. So at the bottom of the slide, you can see a comprehensive, a composite view of all the layers across all the index trenchers. And so the excavation was carried across 137 trenches, 5 by 5 meters in size and in area. And they were divided into 17 house complexes, again, very rich in antiquities, including seals, ceilings, and the regular repertoire of Harappan material culture. So the early Harappan comes from the northern part of the site, marked here in blue. And then that is where the index trench 3 was also taken. And then we have early mature transition coming to us from the southern or the southwestern part of the site. The rest of the area, they have very disturbed upper deposits, the surface layers, which I did not examine. The mature Harappan context comes slightly below the surface areas. And then we also have a pottery kiln near the site. So these are all the different house complexes that I have marked here belonging to the mature Harappan period, divided into 2 phases, 2A and 2B. So the 2A period is marked in purple color and 2B is marked in red color. And so each house complex had, if you see the map here, it had a central courtyard kind of an area surrounded by many small and medium-sized rooms. There were also small lanes that were bifurcated. The main street was found here. And there's a drain here, which a high accumulation of remains. And each of these house complexes had kitchen areas with chulhas, storage pots. But unfortunately, many areas of the site were disturbed by later period pits and garbage dumps. So those were not included in the analysis, but more or less the rest of the area was analyzed for its faunal remains. And so at this site, I took a slightly different approach. I decided to look at the faunal remains in a very micro-contextual way. So, for instance, the bones that came from, let's say, this particular structure or this particular structure, they were analyzed separately, and they were given different unit codes along with the name or the nomenclature of that particular structure. So this helped me to understand the context of the remains much better. So for instance, if I was analyzing bones from the drain, I would expect them to undergo different types of modifications when compared to bones from the street or some other garbage area. So the methods were very similar to what I had done in Europe as well. On the side, there's a lot of sorting, washing, cleaning, labeling them into bags, and then photography of the bones, both in situ and in the bags. Then once I brought the remains to the lab, I identified the animals for their taxa and their anatomy. Then I recorded many different variables like their fusion, the side of the bone, whether it's from the left half of the body or the right half of the body, the portion of the skeletal element that has survived, taphonomic signatures like human marks, carnivore marks, or any natural agents. And then finally, I conducted different types of statistics and quantification on them, and I'll talk about that now. But I'll mostly be restricting to the wild animals. And as expected from any Harappan site, there were lots of domestic animals like cattle, buffalo, sheep, goat, and pig. And we already know that Harappans use them for many different primary and secondary purposes like meat, dairy products, for transportation, and such purposes. But we already know a lot about them. I wanted to concentrate more on the wild animals and see what they can tell us about the Harappans. So in Girawar, there were 196 bones out of 2,449 bones. And there were 53 mammals in them, and the rest were shells and fishes. So these are some animals that we are all familiar with, and I did not want to give you the scientific names of them. So I've given the common names here as well. We are more familiar with the common names like nilgai, spotted deer, black buck. There are wild pigs, the chinkara or the gazelle. There's common house rat. And then surprisingly, in Girawar, we have the wild buffalo as well. It's not just in Girawar. Wild buffalo has also been identified in some other Harappan sites. And we have a few freshwater shells here. And Siluridae is a type of a catfish, which is also very common and found ubiquitously in many Harappan sites. So we have few wild animals when compared to Farmana. In the early phase of Farmana, we have all these different wild animals. And for example, hog deer, there's mongoose, wolf, and the elephant, mainly in the form of the ivory. There are also birds and this reptile, the turtle. And besides that, we have porcupine, house rat, hare, and certain shells. And so these are ubiquitous. They are found both in the early as well as the early to mature transition phase. But when we come to the mature Harappan period, we see a kind of a boom in the diversity of species. So in the Harappan period 2A, we find black buck, wild cat, sambar, four-horned antelope, goose, and some of these shells. This is the kauri and another type of fish. And the ubiquitous species, ubiquitous in the sense they're also found in other Harappan sites as well as in this region. They are nilgai, spotted deer, peacock, etc. When we move to the next mature Harappan period, that is 2B, the diversity again increases multifold. So along with these ubiquitous, we find the Indian bison or the gaur. There's barking deer, mouse deer, gazelle. Then we have tiger and palm cat and mongoose and porcupine. So different types of animals appear on the fauna list. And so very clearly, as we move from the early to the mature period, it seems as if the inhabitants were exploiting or interacting more and more with different diverse types of animals. So, but it is important to know that we have to keep in consideration the fact that the excavations at both these sites are just sample. The entire sites have not been excavated. And even out of the entire material, faunal material that was available, we have sampled some layers, or we have left out the upper deposits, and we have mostly concentrated on the lower layers because they were more primary or less undisturbed. And so these animals are only representative of those excavated layers. And there could be other problems. For example, even though I'm an archaeologist and I've been studying animals and their remains, I could have failed in identifying some of these bones as wild animals, or I did not have the reference collection of that particular species with me, and so I could not compare it with anything and identify it, or they were so badly damaged that identification was not possible. So at Girawar, we see that mostly we have a lot of fishes, and then we have hares. There are a lot of shells, but the other animals like pig or wild buffalo or deer and antelope, they are less in number. So it seems as if aquatic sources were more preferred by the inhabitants, but this again depends on how well these remains are preserved, and they're only representing that part of the site which is excavated. So they have a higher representation, the aquatic creatures, in comparison with the terrestrial wild mammals and non-mammals. And there are no reptiles at this site. However, in Farmana, we see a slightly different picture. Small mammals are dominant in all the cultural periods, early Harappan and mature Harappan. The deer are better represented in the early Harappan period. Antelopes are represented in all the periods except the early Harappan, a contradictory scenario. Then we have wild pig, which is represented in all the periods with variation. Canids, by that I mean dog, for instance, or wolf, they're only present in they're present in all periods, but the cats, like the domestic cat, for instance, or the wild cat, is only present in the mature Harappan phases. Birds, fishes, mollusks are present, almost all of them. And the fishes, birds, and reptiles, they have proportional distribution, but the mollusks are higher in the early Harappan and they decrease in the mature Harappan period. What it means is probably a very direct correlation or interpretation would be maybe shrinking of water sources or dependency on other types of animals, or it's just that part of site which was excavated did not have those remains. So coming to the wild animals at Girawad and Farmana, I first wanted to map them on the site. Where are they coming from in these complexes? So very clearly, all these wild mammals are present or concentrated mainly in these complexes like 6, 8, 7, 4, and 3, and the others are slightly sparsely distributed. So here are some of the remains of these animals. These are the foot bones of nilgai, and we have the long bones, pelvis, and horns of the black buck, and we have the long bone of gazelle. The non-mammals are also concentrated in this central part of the site and in those very complexes where the wild mammals were also found. And here we have examples of fish bones, extremely charred, and shell remains. Now that we know where they're coming from in terms of their presence and absence, now I wanted to understand what are the, how many fragments are present across the site. So clearly, majority of the wild mammals, they came from the garbage or the dwelling areas, but the rest of the pits had very few remains of these wild mammals. So now that I know certain areas have higher concentration and certain areas have lower concentration, I wanted to understand what type of bones are these. Are they bones which are rich in meat, or have no meat at all? So I use this Earpman ratio, which basically tells me the presence of high meat-bearing parts versus low meat-bearing parts in terms of, so C is denoted as low meat-bearing parts, A is high meat-bearing parts, and B is medium meat-bearing parts. And if I take a ratio of these three in terms of the low meat-bearing parts and plot them across the site, I would see three scenarios would arise, and I would see that C is equal to 1, that is low meat-bearing parts are the same as the high meat-bearing parts, or low meat-bearing parts are lower or higher than the high and medium meat-bearing parts. So areas which had greater concentration of skeletal elements, they had high or medium meat-bearing parts like garbage areas or dwelling areas. Low meat parts are not present at all, and if they are present, mostly around the pottery kiln or the storage and some garbage areas in other complexes. And there is no specific area for garbage disposal or butchery of animal remains. And so coming to wild animals at Farmana, again, we see a very haphazard distribution of remains across the complexes. So deer remains are present here, shown with this gray shaded areas, and antelope remains are present in the green shaded areas. And these are some of the remains of the animals, deer and antelopes. Similarly, small animals and, for example, even nilgai, they are also present in very haphazardly across the site. There's no patterning as such within the site. For instance, felids, the cat is present over here. Small mammals are more in concentration in the central part, and there are very few remains of nilgai on the eastern side. And so here are some remains of these animals. These are nilgai bones. Some of them are charred and cut, and they show anthropic modification and also post-depositional modification. These are bones of the cat, of the hare, and bones of the porcupine as well. So non-mammals are also randomly distributed. There are bird remains mainly in the northern part of the site, and we have fish bones over here, and the rest of the areas, they contain the reptilian remains. Shells are very few. And so the next step, now that I knew where they are coming from in the site, I just wanted to understand how the differential deposition of these wild animals has happened across the site. So if we consider that an antelope or a deer was killed either in the vicinity of the site or very far from the site, how would they bring it to the settlement, and how would the meat distribution happen across the site? So this is a method given by Elizabeth Reitz and Wing, where they have divided a standard antelope or deer skeleton into different body parts, which are rich or not rich in meat. So there is head, axial part comprising ribs and vertebrae, forequarter, hindquarter, forefoot and hindfoot, and finally the foot. So we all know that the foot parts are really not rich in meat, and they are, if you go to any butcher shop, you'll see that they're immediately discarded, although some people like to use them to make soups and other things. But generally, it's the forequarter and the hindquarter which are very rich in meat, axial part, and sometimes the head is also brought back because maybe they want to utilize parts of the head or the foot as raw material to make objects or tools, or maybe they want to use the skin of the animal for certain purposes. So we could find all parts of the animal in the site, or we could only find certain parts. So that is going to tell us about how their interaction was with the wild animal. So in any given standard deer antelope skeleton, we would find all these parts and the number of bones for each of these parts. And this is what we should expect from a site. And if we compare this now with what we get from the archaeological record, that will give us an idea of what has survived, what has been exploited, and what has been discarded. So at Girawar, when I plotted the log values of the archaeological remains versus the standard deer or antelope remains, I saw that there seemed to be some kind of distribution of these wild animal meat or body parts across the complexes. I cannot really say if the inhabitants were specialized, were hunting, and if all the complexes were involved in hunting, or were they interacting with a group or community living outside of their settlement who specialized in hunting. But definitely, occasionally they were killing wild animals and bringing them to the site. And in most cases, the differential transport of body parts, that is the skinning, disarticulation, and the processing of the meat happened outside of the settlement, because we do not find all the body parts of these wild animals within the site. So the non-meat parts are definitely missing from the assemblage, whereas the meat-rich parts are much higher in number. There seemed to be some kind of distribution of meat between the complexes. For example, 7, 8, 9, 11, and 13 had very little or no high meat parts, whereas complexes 1, 2, 3, 4, and 6, 5, and 6 had some kind of meat sharing activity between them. So this is, again, considering that the bones are coming from exactly where they have been deposited, and there is not much of site formation process to displace them into the secondary context. At Farmana, a different scenario emerged. It seemed as if the animals which were hunted outside of the settlement or were killed if they had accidentally strayed into the settlement, the entire body was brought into the site and it was distributed, because we have bones of all body parts, right from the head to the toe. Again, we cannot really say if all the complexes were indulging in hunting or they interacted with other specialized communities outside of the settlement, but there is no differential transport of the animal carcass. And we find all parts of the body, and there was some kind of meat distribution going on between the different complexes. So with this, I just wanted to, with these two case studies, I just wanted to impress upon you that depending on the kind of research question we have and the kind of data that, or the remains that come to us, and once we start analyzing them, we'll realize that the kind of methodologies that we began with initially would probably not be enough to understand or to tease out those different nuances of human-animal or human-wild animal relationship. And for example, in Isenia, it was not enough to just know what are these animals and how they were exploited, but it was also important to understand if that was in fact the primary context site, that is where the activity of animal butchering was happening, and what was happening to the remains after the humans had left the site or had abandoned the area, or probably the other natural elements had acted on the site. Similarly, in case of Girawar and Farmana, it was important to understand that even though these wild animals are very few in number in the entire faunal assemblage of, say, thousands and thousands of bones, they are just a handful in number. But if we find out the right kind of methodologies, appropriate methods to employ, and if we ask the right questions and we are flexible enough to modify our research and our methodology and our questions as we go through the process or the analysis, we would be able to find out more and more interesting things about these relationships. So I think archaeozoology is a subdiscipline which allows for this flexibility, although we heavily borrow from zoology in terms of principles and methods, but we can always take the help of other techniques or methods or tools like statistics or quantification or even GIS to better visualize and interpret and understand our data. So with this, I just wanted to end the talk. And at the very end, I would like to thank the Archaeological Sciences Center, the Department or Discipline of Humanities and Social Sciences, and my host institute, IIT Gandhinagar, the excavation teams at all these sites, comprising students, locals, sometimes even tourists, and professors, faculty from different institutes who came to the site and helped to unearth these faunal assemblages at Isenia, Lazare, Farmana, and Girawar. And I want to thank my supervisors, Dr. Joglekar from Deccan College, Dr. Patricia Valenci from Natural History Museum and the National Center for Research, Scientific Research in France, and also my other supervisor, Dr. Ursula Hohenstein from University of Ferrara and the Center for European Research in Prehistory. I wanted to thank the excavation team at Lazare as well, because they helped. It's another site in France, but they helped me to excavate better and to analyze the faunal remains much better. And finally, Pankaj Goel, my colleague from Deccan College, and Giuseppe Lembo, who helped with the GIS analysis at Isenia. So yes, I did keep with the time, more or less.
Thank you. Thank you so much for this wonderful talk. It has really helped to understand the other dimensions of archaeology. I mean, normally we do the cultural remains. We discuss a lot about the cultural vestiges, but there are different aspects which we need to understand to have a holistic understanding of the ancient past. So there are a few questions. I will take it one by one. The first question is by Devpriya Khandelwal, who asks about, why did you select a Harappan site for the study and not a site contemporary to Isenia in the Indian context?
That's a nice question. Well, come to think of it, so I studied the Harappan sites as part of my PhD at Deccan College, and I chose these sites mainly because the excavations had just started when I registered for PhD. And all along, I had been looking through the work of Professor Joglekar and some others, that they were mainly looking at sites which had been excavated before, or through conversations with them, I understood that it's better to analyze the faunal remains from a site that is being excavated. Okay, so that you can participate in the excavations, you can see where the bones are coming from, you can for yourself document and record all the contexts and the little changes that you see, and even kind of teach or train the people who are on the site to sort better, to pick up the remains more carefully, to bag them carefully, record the context very carefully, because the kind of work we do is mostly post-excavation, right? We bring the bones to the lab and then we analyze them. And a lot of information goes missing because they have been recorded by someone else. And had I not participated in the excavations at Farmana and Girawad and had analyzed remains from a site excavated much earlier, then I would have not had these, important intricate details of the context, for instance. So being on the site helped a lot. Firstly, that was the reason. And I could kind of micromanage many things about the faunal recovery of faunal remains, their recording, their documentation, and post-excavation analysis. Also, I site contemporary to Isania 5 lakh years ago. I don't think it's there in India. And I would love to look at the faunal remains, say from Isampur or from any of these lower Paleolithic sites, because I now understand why they would look like the way they do or what kind of methods to employ there. But I don't have access to them currently. So maybe someday I would love to look at the prehistoric sites from India. But I would always go for sites which are going to be excavated or are currently being excavated. So it gives you a very different perspective.
Yeah, I was also about to say that, I mean, we are not lucky to have, such ancient sites which has in situ faunal remains as well as tools. Had it been there, I think you will be really interested to study them. The next question is by Professor Ajit Prasad. He asked, there are a few fish remains from the 2 Harappan sites. Were there any otoliths recovered from the 2 sites? If they are not recovered, could be due to biases in collection method?
Well, that's interesting because at Farmana, because I was there on the site through all the seasons of excavation, even at Girawad, I made sure that every small faunal remain was picked up and bagged. And especially the fish remains or shells. And interestingly, as in the index trenches, as we moved from the uppermost layers to the more deeper layers, there were more and more and more fish remains coming. So as we reached the lower parts of the mature Harappan, or in some areas it's claimed that there is early Harappan because of the pit dwellings, there were more fish remains. And I could see it as the soil was being sorted, and I made sure that they were picking up everything, but there was no otoliths. As far as I can recollect, there were no otoliths. I don't know why it is so. And most of the fish remains were charred. And the problem with fish is that it's very difficult to identify these skeletal elements. At the most, we can just macroscopically, we can say that, okay, this belongs to fish, but cannot really come down to the specific taxonomic identification or anatomical identification. Otoliths, I don't know why they are not there, even in spite of having such careful recovery.
Here, the next question is by Shikha, who asks, Ian Hodder has talked about animal hunting playing an important role as ritual symbols. How can archaeologically we understand these dimensions?
Animal hunting as?
Animal hunting played an important role as ritual symbols.
In what sense? So you're saying there's kind of a ritualistic aspect to hunting?
Yeah, maybe.
Because it's also a conjecture, especially from the prehistoric contexts, lower Paleolithic, middle Paleolithic sites, that hunting was an activity that brought humans together, that created this band formation or group formation, and probably even led to the genesis of language, evolution of language or communication between these group members. Because hunting, if you look at hunting, it's not just an economic activity where you're killing an animal for its economic uses, like meat, marrow, grease, bones to make tools, or the skin to make clothing. But I think I don't know, but maybe over time it would have probably had some kind of spiritual or ritualistic connotations as well, because there are, through ethnographic parallels, we know that there are communities who who pray or who worship before they go for a hunt, or they don't consider hunting as just hunting. And they look at it as a group activity where they bond, there is cooperation, they in fact pray to the animal before they hunt it, right? They kill it. And then once the animal is killed or hunted, they again conduct some kind of rituals, or they pray to it. They apologize to the animal for having killed it, and then they consume its meat and other parts. So is it in that sense that you're asking me this question? But I'm not very sure about this. Is a very abstract concept that is not going to be very directly visible in archaeological records. So unless, they're very definitive exclusive scenes depicted on cave art, for instance, or rock art, where we see humans showing some kind of a ritualistic aspect to this activity. All I can think of is right now is this, that because see, they're not just hunting small animals that they can very easily overpower and outwit them. They're also hunting very large animals, and in which case they could themselves become the prey for these large animals, right? Easily. So I think there would have been some sense of awe or fear in them when they would kill such large animals, and there would be some belief system in place before taking up these activities or at the end of these activities. But apart from that, I cannot speculate anymore, I guess.
There is one more question by Shika, who asks, in Gujarat, there are pastoral communities who don't consume animal meat. Do you feel that every society treats the animals differently?
Yes, of course. And it's an evolving relationship, right? It's a very complex, evolving relationship. Even us, people who live in urban areas, we are undergoing this major dietary shifts. Many of us are becoming vegan or adopting vegetarianism. I'm sure there are communities who have these dietary preferences. And so, sorry, what was the question again? Communities in Gujarat.
Do you feel every society treats animals differently?
Yes, they do. They come from a very different, geographic region, different climates. Their interaction with animals will be very varied and complex, and it will evolve not just over a long period of time. It may evolve even within one's own lifespan. So I'm sure it is very, very, different from region to region. But it's a very vast question. I mean,
yeah, and it's also difficult to find it archaeologically. I mean, how
yes,
it's an intangible aspect, maybe.
Yes.
Yeah, there's a question from Jay, who asks, have we found remains of domesticated dogs or cats at any of the Harappan sites?
So yeah, so there are very few in number, but if you look at these remains, they do not have any kind of cut marks or charring marks or any sign of human modification on them. So we want to tentatively interpret as, these animals were just kept as pets and they were not really consumed. And this can be corroborated very well with, say, there are terracotta figurines of dogs, right, from the Harappan sites, and some of them even come with collars around their necks, which even kind of substantiates this belief that they were kept as pets. Cats, I'm not so sure, but yes, dogs, yes. There are also, what kind of evidences do we have? I showed you a pottery piece with paw marks on them, paw imprints on them, which shows that dogs were definitely around the human settlement. These are commensal animals in the sense they benefit from being close to human settlements. This is a kind of a symbiotic relationship, and they wherever there are humans, there will be dogs, there will be crows, cats. These are animals that get shelter, some sense of security, food definitely from the humans. And humans also have something to gain from them, companionship. They are maybe they help to relieve stress sometimes. They are used as assistants for therapies. They could be used to keep other animals out of the settlement as guard dogs, for instance, or they could be taken to the fields to keep away these other animals like antelopes and deers, which were treated as pests. So probably as pests, those who destroy crops. So I'm sure these animals, and they have a very long history of domestication. We know that dogs were one of the first animals to be domesticated. They've always been around human settlements, and they were not consumed. That's the evidence from all the Harappan sites or any other site. They were not consumed.
The next question is by T.S. Subramanian, who asked, there is a school of opinion that the fortification walls in some of the Harappan sites were meant to ward off attacks by wild animals. Do you agree with that view?
Well, could be. I know that most of them, most of the we believe that Harappans were non-violent. They did not really indulge in wars or conflicts. So why were these fortification walls put up? That's one of the biggest questions regarding these structures. I'm not so sure. Yes, if there is a wall, if there is an obstacle or boundary, it's going to keep the animal away. But the animals are very smart. They will always find their nooks and corners to get into the settlement, especially the small animals, small mammals. But about large animals, yes, probably. I mean, they could have been, the walls could have been erected to ward off these animals as well. But I believe that even if these animals did stray very close to the sites, they could have been hunted or they could have been shooed away. Not every animal that strayed near the site would have been hunted or consumed. For example, we have remains of leopard and wolf. So this is a one-off incident where maybe someone spotted it near the site and probably killed it, or some other animal killed it, and their bones have got included into the assemblage. But they were not, I'm not sure if they were hunted or they were consumed, or what would be their value or purpose in the lives of these ancient inhabitants.
Professor Ravi Korisetter wants to ask a question. Sir, are you there? You can ask.
I don't know whether you can hear me.
Yeah, we can
hear you.
Yeah.
Okay, I would like to supplement. One thing is, in the Southern Neolithic, we have a package of animals which are domesticated. The dominant one was cattle. And of course, you have sheep and goat and other subsidiary animals. The faunal analysis has revealed that except cattle, other animals were consumed. There's no sign of any kind of cut marks or charring or calcined feature, associated with these boss remains. So it was a ritual animal, as you have seen. Both of you have seen. We have cattle, especially the bull, having a special place in the Neolithic culture of southern India. So that kind of ritual relationship is always there. It is considered a male fertility deity, and it can be traced back to these pre-pottery Neolithic in Southwest Asia. There are these sites like Murebit, Huriya. So I can't pronounce these names, but there are those sites which have clearly revealed the evidence of cattle, assuming an important place in the culture, and was not consumed. Is a male fertility deity. And that is one reason why the rock art landscape at Sanganakalu has numerous imageries of cattle depicted on the landscape. And there is something directly related to fire cult or fire cult and cattle domestication. Maybe there were these animals among some societies which were, not consumed at all.
I think that's very important, because
make a supplementary point. That's all.
That's very important, because we should not just report our findings in terms of presence or absence of animals. We have to actually look for those marks in them and then make those claims that they were consumed or not consumed.
Because it was described as an agro-pastoral society and pastoralism dominated by cattle domestication and so, but it was not an animal which was part of their diet. as we understand so far. I mean, if you come and look at these bones once again, maybe there'll be something different coming
Because
up.
many of these marks that we attribute as anthropic or anthropogenic, they're very faint and they're very tiny. And if we are sorting through bones, very quickly, and we are just recording certain variables, they will get missed out very easily.
If the bones were split for extracting marrow, you will see a different cut mark.
The breakage is very typical in case of human induced. Yes. Also, I think the age of the animal, so the animals would have eventually died at some point, right? These cattle, but not of human causes, but natural death probably. So I think doing a kind of an age profile of the animals would also help. Because if we have most of the cattle living beyond, say, 10 years, 14 years, then we can definitely say that they were not being killed.
Was it the case in the Harappan context, cattle was consumed? Cattle meat was consumed?
So it is interesting because in some sites, for very few sites, we have this mortality profile, this kill profile, right? And for example, Laura Miller's work at Harappa, or say Ajita Patel's work at Dholavira, they very clearly show that animals of different age groups were there. Females, for instance, were kept beyond 7 years, 10 years. And most of the, in Dholavira, for instance, we have a lot of buffalo, but more mature buffalo, mature cattle.
This term cattle is generic, isn't it? It includes both buffalo and cow.
Yes, but also the proportion could indicate something. We don't know. Buffalo has more, to offer in terms of meat and dairy compared to cow. So if we do the age profiling and shows that in some sites they were deliberately keeping some of these animals alive for a very, till their old age.
Full life,
And probably they would have preferred sheep, goat over cattle for meat, or but it's very patchy evidence. It comes only from 1 or 2 sites. In my case, I did not do the number of individuals for Farmana and Girawat because the bones were very mixed. They were from very close contexts. So there could easily have been an overestimation of number of individuals and their age groups. So the bones could have actually accounted for just 1 or 2 individuals. But because they're so broken and they come from such different contexts, I could have easily gone wrong with the calculation and said there are 10 cattle. Although, there could just be 1 or 2. So I avoided that quantification because it could be highly erroneous. But that would have told me this kind of, data, that how old they were, why were they surviving so long, and what could have been the purpose behind it. So
it is generally suggested that this, especially cattle, was domesticated independently at 3 centers in the subcontinent. But what we see in our record is in a domesticated form, it appears. We have not seen the earlier stages of transition from wild to domestication and so on. So pastoralism as such was introduced into Southern Neolithic province. That is one conclusion. And the second conclusion is that these sheep and goat, they adapted to all kinds of diverse environments, arid, semi-arid, and all kinds of favorable congenial.
They're easier to manage.
Manage. So they were the main source of animal protein and not the cattle. This is the kind of inference we have so far.
And cattle have very varied uses. We can take them to the fields. We can yoke them.
Dot animals,
very, very useful for other purposes. And pig or sheep and goat have fewer economic uses.
Thank you.
Thank you, sir. We have a question by Camelia. She asked, so much to learn. I wanted to know about how the cultural relevance of human-animal relationship can be deduced from these 2 case studies. Like, are there any distinctive cultural aspects that still prevail in contemporary times?
So culture in this sense can be, so we are interpreting it only through these physical remains, right? And it is not actually enough to interpret them only from the faunal remains. I would say that in order to understand, for instance, in Isenia, if I really want to understand the culture of these hominins, human ancestors, what are the things that I can deduce from this? Just from animals, it wouldn't be enough. Ideally, I should look at the data set of fauna along with the data sets of lithic tools, let's say, or the climate data, the plant data, and bring it all together and then talk about why humans would choose a place like that. How many humans could possibly have lived there? And why did they develop the kind of lithic tools? For instance, this is a lower Paleolithic site, and any student of archaeology will say lower Paleolithic site, and it should definitely have hand axes and or bifaces and cleavers and large tools and all that. But at a site like Isenia, you don't. We have very small tools, small flakes, sharp edges. They are called denticulates, notches, little little tools with very sharp edges. So why were they using such tools? And why did they create such an assemblage to process the animal meat? Why did they kill only these kind of animals and not the others? Or why would they? How would they exploit, say, animals which live at higher altitudes, like the tar, for example? There's an animal called tar, and it probably came down to the valley side only in a certain season, but not throughout the year. Bisons, instead, they would cross through the valley in large herds. And so these bands of humans, you would require not a few humans, you would require at least a good group of 30, 40, 50 humans to be able to charge and attack such large groups of animals, kill them with the kind of tools they had, and then decide how much of the meat to bring back and distribute that meat among them. And so these are, small decisions that are made probably with some kind of communication between these groups. And that is the culture that you can think of. Culture is, what is culture? It's a way of living, isn't it? Our belief systems, our traditions and customs, whatever. So at a prehistoric level, this is all that we can interpret. Why would they live in such an environment, and how would they optimally use it to their advantage? At a protohistoric site, it would be very different. When we already have domestic animals, why would they want wild animals, for instance? And if they lived in such large settlements and they had crops and domestic animals, why would they distribute meat to others? If I live in a certain household, in a certain complex in Farmana, and I go out and I find this antelope and I hunt it, and I decide, okay, I want to eat its meat today, I bring it back to the site. Why would I distribute the meat with my neighbors who are living in the other complexes? So these are questions that I think would come under this broad idea of culture, and try to address them, but it's difficult to with very limited evidence. But I think this is what we should try to aim to address in different contexts. As to the why is very difficult to answer more than the when and how and who. Why would they do this? Why would they not do this? And culturally, how did they evolve with their technologies? And survive these different, climates or environments. So I think that is what comes to my mind right now. Culturally, I mean, if someone else has something to add. It's a very broad question to address, very complicated, because culture is such an all-encompassing word, right? Everything is culture. So why certain animals were killed and not others? Were there taboo systems in place? As someone asked, were there rituals for these animals? It's very difficult to answer these questions.
There's a question by Apurva who asks, what do the seals containing animal imageries suggest about the trade?
Suggest about the trade? Okay, two very different concepts that have been brought about the Harappans. Well, what I have understood from the seals and sealings and their variations across the length and breadth of the civilization is that not all animals are depicted in all the regions, right? There are certain animals which are common throughout the civilization, like say unicorn, for instance. But there are some animals which they're depicted on seals only from certain areas or certain regions. And although they have chosen to depict very wide variety of animals, you will find even there's depiction of an ass on a seal from Kanmeh. There are elephants. There are, of course, the zebu, the cattle, the humped and the unhumped cattle. But there are also creatures like the unicorn, mythical creatures which we don't know how did they think of it and why would they depict it on the seals. In terms of trade, I'm not going to correlate it with that and make this far-fetched statement that seals were used to trade only those animals, and they were the animals that were being traded were depicted on the seals, or maybe the goods that were being traded were sent with those on those animals that were used as transport for transportation. That would be a very far-fetched thing to say, but I don't know if there really exists any correlation between the two. They have chosen to depict certain animals. Maybe they were more symbolic in nature, or there were animals that they revered or worshipped or had attached some kind of socio-cultural economic significance to them. And they acted something like your barcodes or like a mark, you know, a certain trader or a merchant's mark, and were put on those seals. But beyond that, I don't really see any correlation with trade.
Yeah, SK Aruni, he has some pointed questions. I mean, whether the history of animals is only restricted to remote periods like prehistoric or protohistoric. What about the historical or medieval period? What about the evidences from the historical and medieval periods, and how they are studied?
So I have not personally studied any historical medieval site, but I have gone through some faunal reports. And what I understand is that for such sites, we are actually at an advantage as an archaeologist, because we don't just have the bones as evidence. We also have different corroborative sources. We have the texts, we have iconography, images on temples, for instance, or other structures. We have a lot of paraphernalia, artifacts which depict these animals, and so we can very easily fact check or cross check and interpret. And we also have other things like paintings and sculptures and other things. So we have a lot of data. I think we have much more data. Archaeology is a very evidence-based discipline. So are other sub-disciplines of archaeology. But archaeology is if we go further and further back in time, it becomes more and more difficult to practice because we are left with just the bones at some sites, and we have nothing else to corroborate them with, and we just have to go with whatever methods we have developed within the field of archaeology, which is mostly, say, macroscopic or microscopic. And now we have a few other methods like isotopes and maybe DNA or proteins. So we are very limited in terms of our approaches or methods, and that's why the interpretations are general or very few. But when we come to the other time periods, we should not be facing such conundrums. We have so many different sources to look at to better interpret our assemblages. And I think more and more sites, historic and medieval, should be studied, and different aspects of these relationships should be brought to light. But definitely, I don't think our relationship with animals began at 2.6 million years ago, or 3.3 million years ago, and at Lower Paleolithic. It's a relationship that's very primal. It's primeval. And it probably goes back much longer. So very broad answers, but the questions are really broad.
But always, I mean, there is no restriction for, studies during the historical and the medieval period, and it will really supplement whatever historical.
Whatever we know, yeah.
Yeah, the next question by Saurav Lai. ancient people created tools by their own for hunting, and these were various types. But at Nevasa, stone tool making factor was discovered.
Okay.
So any kind of evolution of the tool making process, I'm unable to understand this question.
Okay. The question ends there, or there's more?
Yeah, that's it.
So tool making is again osseous artifacts, as they are called, is a completely unexplored area in Indian archaeology. It's something that is craving for attention, craving for multidisciplinary studies. And there's a lot of scope there because we have amazing sites with amazing evidences of tool making or ornament making. Shells have received a lot of attention. So we know what the process was, where they were acquiring their raw materials from, which are the shell manufacturing industries, which are the sites, because we have, the raw material, the debitage, and the final products. And the same kind of studies have not been done for, say, ivory artifacts or bone artifacts or artifacts made out of tooth or some other part. So that is something that requires immediate attention and a lot of scope to study there. And because many of these tools that we know of from in the protohistoric period, needles, awls, stylus, whatever they are, beads, bone points, for instance, they find their origin much before, right? We have tools coming right from Mesolithic, Neolithic, in even Upper Paleolithic. So, and in European context, much earlier. So there are certain styles and forms that have survived over time. They have undergone very little variation, modification. And there are other forms which have appeared very suddenly in different cultures. And then they also disappear. So this is something that needs to be studied. But if you want to really understand a bone tool industry at a site, it would be difficult because, see, when a bone is broken and something is fashioned out of it, and there are lots of wastage, like we have shell wastage. Similarly, there'll be ivory wastage or bone wastage, but it will be very difficult to discern it in the assemblage, because a bone broken by a human, a bone broken by a carnivore, a bone broken for making a tool, they will all leave very similar byproducts, secondary products, the wastage. So as an archaeologist, when I'm looking through bones, the fragments of it, very difficult to say that this particular bone is a result of an artifact manufacturing process, unless there is some, telltale evidence, like a proper blank has been produced, there is some mark on it, some incision has been made, or it looks unusually straight and the dimensions are very, unique, and the breakage pattern is very different. Then I would say, okay, this is a bone that was in the transition, it was in the process of being converted into a tool. Otherwise, it's very difficult to discern, unless if they're found in the same activity area, in the same zone. The final product is also there, the raw material is also there, the different stages of manufacturing are also present. That would be very difficult to discern. So I'm not very aware of this Nevasa bone tool industry, something I should look into.
There's a question asked by two participants regarding horse. The most controversial.
I knew this question would come.
One is asking, Shika asks your opinion on horse debate, and another by Gauri Badekar, who asks
my friend,
observations on horse in Harappan culture and nature of use of horse based on faunal evidence.
So there are a couple of points that come to my head immediately when I think of horse. I believe that by the time we come to the Harappans, they knew what animals they wanted to interact with and in what, way. Harappans lived across very different ecological zones, right from, Haryana to Gujarat to Sindh, Pakistan, and Maharashtra. You have such variations in climate and, the biomes or ecological niches. So in different regions, we see that they are coming in contact with different types of animals, and there is some kind of interaction. Either but the interaction is primarily based on the value that that animal would have for them. That's what I really believe. When it comes to horse, the first problem is with identification. So the way cattle and buffalo bones look very similar to each other, or sheep and goat look very similar to each other, horse or equid, the family they belong to, horse bones are very similar to ass bones. Or we have another species to complicate the whole matter, that is the onager, right? Equus hemionus. So there are three different animals, three different species, which all look very, very similar. And we really don't have a very good laboratory or a reference collection in the existing departments of archaeozoology all over India, where we have all the three animals, complete skeletons of male and female and of different age groups, a young one, a subadult one, a mature one. So that if we were to find, tentatively, if we were to find these bones in the site, we could immediately take them to these labs and compare them and come to some conclusion. But that is missing. So most of the interpretations are done by those people who have had some expertise on this. I don't know what they have consulted, actual collections or just published illustrations. For example, Richard Meadow or Bocconi or Sharma, they have all given their own interpretations, but not just based on morphology, but sometimes also based on measurements. So that is one problem. We do not have enough measurements. We do not have enough comparative references to say. Second thing is the context. Very rarely have I seen description of the context from where the horse bones are coming. In my own experience, Gauri was there with me in Farmana. I did find equid bone from a pit in Farmana, and I was very confused. Should I report it or not report it? At the end, I decided I'm not going to talk about any of the pit remains, because pits could be of any time period. They are extremely mixed in contexts. Also the Bok remains. So I did not report them. And I'm sure this was some kind of an equid bone, but went unreported. But those that have been reported from other sites, we definitely have conclusively identified horse remains coming mostly from the later phases of the Harappan civilization. Mature, yes, but also more of the late Harappan sites or post-urban Harappan sites. So I'm sure that, but the remains are very few. So I'm sure that these animals probably did exist, because we do have, horses were domesticated even way before that. And there are, I mean, why would they not know about an animal if the people were living in such varied ecological zones? I'm sure someone somewhere would have come across this animal. They were also traders. They were moving across the continent. They would have seen that animal in some other part, maybe Central Asia or other regions, if not within the Indian subcontinent. But the bones are there, so probably they did know of this animal, but I'm not sure if they exploited them to the extent the other animals were exploited. Also, if you see, most of the horse remains, I think, also come from coastal sites, Kuntasi or say Surkoteda, for instance. There are bones from Harappa as well. They come from certain regions closer to the water bodies. So what role could these animals have played? Very few, one or two individuals, for the inhabitants living on such sites. That is something that needs to be looked into. So I think they knew about them, but they did not use them to the extent that they used other animals. So that's my take on this. Because I have seen equid bones. Currently, I'm looking at the Ulavira remains. There are equid bones in them. But I myself cannot say what it is. It's ass or horse or onager. But they exist. They are there in the faunal assemblage.
So we have two last questions. One is by Jay, who asks about any birds that were domesticated or used for food during the Harappan period.
There are lots of chicken bones, lots and lots of chicken bones. So yes, they knew about chicken and definitely consumed them. There are other birds too, peacocks and pond heron and cattle egret. But they are very difficult to identify. They break very easily. They are they get missed very easily. They're not picked up from the sites. So at the end, we are just able to say what are these different birds, but very rarely have we seen any anthropic modifications on them, and we cannot really comment on whether they were pets or, we can just maybe talk about their presence, absence, and also if they are migratory birds or local birds. But among the domestic ones, the chicken, yes.
We take the last question that is also relevant. What are the limitations of archaeozoological studies? That is by Shikha.
It's a very good question. We are limited by many, many things. We're limited by infrastructure, by excavation methods, by our own training at times, by reference collections, by not being able to keep up with this very rapidly growing field. Most of us are still practicing or employing traditional methods because that's what we are trained in, and we are used to studying the remains from a certain culture or a certain time period or a geographic region. So when we confront faunal assemblages from other cultures or time periods, and which require different research questions with different methods altogether, we maybe shy away from it, or we find that we are not adept enough at it. So when I will only talk about myself here, but for instance, I studied protohistoric sites. I was used to seeing cattle, buffalo, sheep, goat. And then when I went to Europe and I had to look at the animals from Italy and France, and they were very different animals. It took me a while to accommodate myself to that scenario. And I had to go through many different collections, read a lot, and then slowly kind of slide into this whole faunal analysis and make myself comfortable or confident enough to analyze the remains. Excavation methods have to be really, really rigorous. We do not do 3D recording of all the remains. Forget bones, even the other remains. We don't do 3D recording of the context of the bones, and we do not recover them properly. We do not preserve and conserve them properly. And so there's a lot of loss of information at the site itself. And then there's loss of information, or these limitations creep in post excavation, where as experts, we deal with missing information, non-availability of reference specimens. We are not trained in certain methods, or we set out with a few research questions and methods, and we are not flexible enough to update them or change them over the period of our study. I think that's also required. For instance, in Isernia, I just wanted to first analyze what are these animals, which species, which bones. But then I realized that there is this larger question of trying to understand who created the assemblage, carnivores or humans. And then I realized, there are so many methods that I can employ, not just taphonomy. There is long bone fragmentation index. There is GIS that could have, aided me in this quest. So that is what is, I think, limiting archaeozoology. And I think this subdiscipline is gaining importance, but not as much as it should. And the last one I can think of is we don't integrate our data set with other data sets as we should, with botanical data sets, climatic data sets, because then a bigger, better picture emerges, and certain gaps may get filled up by other data sets, which archaeozoological data sets may not provide.
One just comment regarding presence or absence of a particular animal in a particular culture context. There was this issue of definitive identification of sheep and black buck. You may be aware of this. Claassen and Thomas thought that there is a lot of sheep in Chalcolithic, Marwan, Jorvay situation. But later Meadows said, no, they are not sheep, they are black bucks and so on. So this kind of issues, but you gave a very circular answer to the question on horse. It's very interesting answer.
I'm saying that the bones exist, but we don't have the right methods.
If you say no, what matters?
Maybe
Uh,
DNA, maybe DNA analysis.
That is the
DNA or
point.
protein, yes, or morphometrics, which the
Yeah.
archaeologists
There
have
is
used
one
in the past.
sophisticated Zoo MS, right? Zooms.
Zooms,
Yeah, I mean,
zooms.
yeah, I recently met one expert who does a lot on Zooms actually. So I
But
will share the details.
as we know, horse was first domesticated in the steppe.
Central Asian
You
region.
see, if you find equids here, we should find some relationship. As he says, genetic relationships can also be, determined. It is possible to resolve this issue, but
So
unfortunately, it is in a
the thing
concealed
is,
way.
yeah, because the horse remains that have been reported from sites, it'll be very difficult for any one of us to go back and go through bags and bags of bones and identify that one particular horse remain and put it for DNA or protein analysis.
Yeah.
So I think it's
No,
a
it could be
time-taking process, but it can be done.
any of the equid family, like Ovis, Ovis in Chalcolithic context.
Yeah, so there are methods and that can be taken
Right.
up, I guess.
It can be now.
Yeah.
There's so much of advancement.
Yes. All right.
Thank you.
Thank you, sir, for your comments and suggestions, and thank you, Sharada, for this wonderful talk and also the well answered Q&A session. There are certain comments also from SK Aruni, really congratulating you and also appreciating the Q&A session. Thank you so much, and I thank all the participants and the panelists also, and also Professor Ravi Korisetter for his suggestions and comments. And we look forward for your presence in the next lectures also. We will be
I
continuing.
was happy today.
Thank you so much, sir. Thank you, Sharada.
Thank you.